Hello. Okay, since these are in PowerPoint, we can actually have subtitles on the bottom. Maybe that'll help a little more. I don't know. Let's see. So let's go through this. We're starting chapter 4, and I would say chapter 4 is probably the most important in the book. We're going to take our time with it to some extent. And this is really the make or break chapter. If you can grasp chapter 4, you've basically got immunology. Everything else is just adding a little piece on to the beginning or end of some process you already know. But this is essentially the way that the adaptive immune system works. And so let's dive right in. All right, so shown up here are some of the things that we are going to cover. There are going to be a couple of these items that are sort of marked in dark blue. Those are not things that we can actually ask a test question on. They're mostly have to do with the structures of proteins and the way they come together. But I've included them so that if you're visually oriented, like I am, then you can see how these proteins come together and how they work. So let's try. Okay. And the nice thing here, which is why I especially like this set of slides from Dr. Schmidl, he has picked out all of the big vocabulary terms. And so you want to make sure you know each of these terms and are able to use it properly in a sentence. So this might be a good case for making some flashcards and going through and just getting all these terms down, immunological terms. I think that's a good way to get into this chapter. And then from there, we will add on the details to it. So, all right, let's start right at the beginning. An antigen, yes, is something that a T cell receptor or an antibody can bind. It's basically whatever the T cell receptor or antibody sticks to. Most commonly, these are going to be little protein fragments, but occasionally they can be other molecules. Or they can be something like a protein plus another molecule that are bonded together or just interacting really tightly. So there's a lot of range of what an antigen can be, and that's good. It means that our immune system has lots of ways to keep us healthy. Immunoglobulins, these are the proteins that we call antibodies, and B cell receptors, which are like a antibody that's mounted to the surface of a B cell. Immunoglobulin is named after a particular fold, like a general way that a protein looks. Think of it that way. And there are actually a lot of proteins. It's really common for proteins that are expressed on the cell surface to have one or more of these immunoglobulin-like folds. And we'll show you what these look like, but essentially if you hold up your two hands in front of you and put them together so that each side is one half of a circle, what you've got there is a pretty good representation of what an immunoglobulin-like fold looks like in the real world. Okay, and so we've got our immunoglobulins, which is the basic fold that all of these antibodies are built from. We're going to call antibodies, we'll call them immunoglobulins. That's just the way that immunologists do it. So if it is a type A antibody, we'll call it immunoglobulin A, and we will abbreviate that as IG, capital I, little g, and then capital A, just like that. And so on. All right, next, B cell receptors or B cell antigen receptors. Yes, those are just the antibody that B cell is going to make, but attached to the surface via a special chain. So instead of one of the chains that would make it one of the MDGE or A type antibodies that we'll get to in just a moment, this has a different chain that's going to link it to the actual surface of the cell. Antibodies are, sometimes known collectively as immunoglobulins, though, like I said, there's a lot of things that have an immunoglobulin fold, like this particular organization in the world. We just happen to give antibodies, full use of this name. For reasons that, now that we understand everything else, don't completely make sense, but that's the way it is. So antibodies are going to be these molecules. You can think of them as in the shape of a Y. They basically have two little ends at the top, like the tips of the top of the Y, these are the parts that can grasp onto antigens, and they're going to bind to those non-covalently, which means they can use all the other kinds of bonding. They can use hydrophobic interactions, they can use van der Waals force, they can use hydrogen bonding. They just cannot make an actual covalent bond. So you can always pry an antibody off of its surface. But not all antibodies bind with equal, well, we'll get to our vocabulary words for this in just a moment, but let's just say with equal strength of binding for right now. And so you can measure your antibodies and figure out whether they're really strong binders or really tight binders, or whether they come off pretty easily. And yeah, there are tests for all of these. And each of these characteristics will affect the way an antibody can be used inside the body. I mean, your body doesn't try to make bad antibodies, but the better antibodies definitely give the immune system more options for things to do. All right, so there are a couple parts of an antibody. You've got a variable region and a constant region that I think is on the next slide. The variable region is what changes in pretty much every single antibody, or at least some parts of it change. And we'll show you exactly the parts. It's kind of like, I don't know, like imagine a hand. So imagine that the central part of your hand, that's the part that's going to stay the same. And imagine that the fingers are variable loops. So imagine you can like change the shape of your fingers. So I'm holding my fingers up here and changing the shape of them. That's pretty nice. That's like what the variable region is. It has a constant core, which is like your palm, and it has some variable regions which help it to get a better grasp on different antigens. So imagine your hand getting a grasp on them by changing the position of your fingers. Only in the case of an antibody, they actually change the amino acids that make up the antibody. And that's how it gains the ability to grab onto different things. Okay, so the variable region is going to be located at the tips, both of the top tips of the Y. The constant region is going to be the stem of the Y. It's going to be basically the bottom of our antibody. And yeah, the constant region is useful because no matter what the tips are binding, the constant region is going to act like a handle. And there are a lot of different cell types that can grab onto the constant region. So we also call this the FC, which is the constant fragment, but we just put those in opposite order. So fragment, constant region, whereas the variable region is sometimes known as the fragment antigen binding, or FAB region. Okay, isotypes are the different types of immunoglobulin, or the different types of antibodies. And so there are many different isotypes. Yeah, to make an antibody, it takes two heavy chains, and they're going to be the same kind of heavy chain. You'll just have two copies. And there will be two light chains. And again, they will be the same kind, but you will have two copies of them to make up a full human antibody. Other kinds of animals just have basically the heavy chain of the antibody. And so we think that's how it probably evolved, like heavy chain first, and then adding the light chain on let some animals have a little bit broader range of things that they could bind. So the light chains on antibodies can be, and you're going to have to get good at Greek letters. I apologize for this because they come up a lot in immunology. They really like their Greek letters, but it's just the alphabet. And so it's not that bad. And I don't know, if you've been in a sorority or fraternity, these may look familiar, except that they're usually going to be the lowercase version of the letters. That's the thing that throws you, right? Unfortunate, but this is a thing that can be learned. Okay, so light chains can be of either, so lowercase k is called kappa, and just like in NF kappa B, right? And this little thing is like a little tent with a hook on the top side is a lambda. So it's like the letter L. So K or an L, kappa or a lambda light chain. And think of it as L for light chain, and K is the letter next to L. So I guess they'd already taken M. You'll see that for one of the heavy chains, so it kind of has to be K. All right, heavy chains. There are 5 different ones, and so these are called, well, yeah. So the first one, it looks like a U with a hook on the side. That's the Greek letter mu, or it's the letter M, basically. So IgM is going to have the mu chain because it's the lowercase m. And it's the same with all of these things. Actually, an IgM will have the mu heavy chain plus either a kappa or lambda light chain. And that's totally variable. Each of these types, isotypes of antibody, can have either of the two light chains. They're both fine. So the second letter that looks like an O with a little, I don't know, hook on the top, that's a lowercase delta. So that's the delta chain, like D. The next one is sometimes drawn as like a little symmetrical Y that goes below the line, or sometimes it'll look like a little fish, but swimming downward. They'll sometimes make a little loop at the bottom of this, like, here, watch my mouth, about like that. And they'll sometimes curl it back over the top to be extra fancy. But in this particular font, that's what a gamma looks like. So yeah, it's not a Y, it's a gamma. The little fish here, that is an alpha. So that's an IgA, and this guy right here is an epsilon, and so that is an IgE, is going to have an epsilon chain. So an IgE will have two epsilon heavy chains and two either kappa or lambda light chains. There you go. And that is the beginning. Now, we'll get to the differences in structure between some of these things. They're quite interesting. The ones that are, let's see, just this simple antibody type are going to be IgD, G, and E. So those are just going to have two heavy chains and two light chains, and your molecule is done, and it's shaped like a Y, and everything's great. IgA is a neat one. It's shaped like two Ys if one Y was pointing up with its two little prongs pointing up, and the other Y was underneath pointing down. And so IgA is able to bind twice as many things, twice as many antigens, as an IgG, or D, or E. So there you go. IgM takes this to just another level altogether. So IgA is going to have 4 of these heavy chains and 4 light chains. IgM, it looks like a star. They're really great. So IgMs are going to be like 5 antibodies chained together in a star, like a little ninja throwing star. And so an IgM is going to have 10 heavy chains and 10 either kappa or lambda light chains. And there's also an extra little chain that joins the IgM and IgA together to get all those parts to link up. And we'll get to those later on. Okay. In the second half of this chapter, that's why I said this chapter is just amazingly huge. We're actually going to get to T-cell receptors because they're kind of built the same way as immunoglobulin antibody molecules. Yeah, to some extent, they work the same way by binding to a little antigen that's usually a little piece of a protein. So, all right, it's a cell surface receptor for a T-cell. So you can think of a T-cell as basically like, imagine evolutionary history. You would start out with something like a B cell that has just heavy chains on the surface and is able to bind things. Now, if you look at sharks, or if you look at things like camels and llamas, they still have this more simple type of receptor. And then you can imagine this cell learning how to let go of its little immunoglobulin. So that would be a neat thing. That would be like the first proper plasma cell releasing antibodies. And imagine a T cell as just like a shortened version of this original cell. It's got something like an immunoglobulin on the surface, and we'll look at exactly what that looks like. And this little short immunoglobulin is able to bind onto antigens, and it's able to recognize other cells that have those antigens on them and then do things like kill those cells or call in extra help. So T-cell receptors are a disulfide link, so that means there's 2 cysteine amino acids, and they are going to be linked together by their sulfur groups. That's found at the edge of the cysteine amino acid. And it is a heterodimer. So dimer means 2 parts. Hetero means they're 2 different parts. And it's made up of a alpha chain and a beta chain. And it's going to be in a complex. So it's going to be stuck to these 2 other proteins. One is called CD3, And so you'll find CD3 on all T cells. It's a general marker. And the other is going to be this little squiggly thing, which is a zeta. That's a lowercase zeta, so just think of it as a Z. And when you get to the end of the Z, instead of just making a nice sort of flat line, you put a little hook on the end. And there you go, you got your zeta. And in some fonts, they'll actually make a little loop up here at the top. But yeah, now you know what you're looking at. All right, so T cells that have these alpha and beta chain receptors are called alpha beta T cells. There is another kind of T cell which has gamma and delta chains, and that's called a gamma delta T cell. And those are interesting. They work a little bit differently from other T cells, and they are present a little earlier in your development. So when you're a little baby, your gamma delta T cells are kind of in charge, and eventually your alpha betas are going to take over and carry you on into adulthood. Okay, now the T cell receptor is just half of the equation. It's one side. It's how the T cell binds to a little peptide, but peptides don't just present themselves. They get presented by major histocompatibility complex proteins. And yep, it has rendered that correctly in the transcription below. And here you can see that word. Histo means like tissue, and so tissue compatibility. These MHC molecules are the ones that people would sequence if they wanted to find out whether you can be a kidney donor or a bone marrow donor for somebody else, for example. And so these MHC molecules are going to be found on, there's two types, class 1 and class 2. Class 1 are found on just about every cell in the body. Class 2 are only found on some types of cells of the immune system. And so that's the difference. But both of these things are going to present antibodies. They're going to they don't present, pardon me, they're going to present antigens. There we go, sorry. So a little piece of protein from inside the cell is going to be loaded onto MHC class 1 and shown on the outside. And a T cell receptor will roll over and clip onto it for a second and check it out. See if it is something normal or something out of the ordinary that needs to be dealt with. Okay. MHC is the major histocompatibility complex, and it's a bunch of genes, and they are on human chromosome 6. So if you want to get on the bone marrow donor list, they will sequence a little piece of your chromosome 6 to figure out what your which genes you have there, which version, because there are many inherited versions of MHC that you could get. But you only get one chromosome 6 from your mom and one from your dad. And occasionally, those two will have the same type of MHC complex at that position. It happens, yeah. And so in that case, you're going to be real limited in the things that you are able to recognize with your T cells via MHC recognition. All right, so MHC molecules are definitely polymorphic, so they exist in a variety of different forms. And so each of these forms is encoded by a different variant of the gene. That's how you get the different forms, and those variants are called alleles. So yeah, a variant of a gene is an allele. That makes sense. And so there are lots of MHC alleles because MHC is a very polymorphic locus, a very polymorphic gene. Let's just say that. Okay, so MHC restriction, is the interesting and terrible phenomenon that tells you that you can only recognize a particular antigen if it's bound to an MHC molecule and if you happen to be able to make a T cell that can bind to the sandwich of that MHC molecule and the antigen that is loaded on top of it. If any one of those parts fails, then it doesn't matter how much of a protein or invader there is inside of your body, you won't be able to recognize it, and it can just grow. So MHC restriction, real and interesting. It determines the way our immune system can see the world. And this is a case, I would say, where people are looking at maybe expanding what your immune system can see. So if you were able to add in a couple of extra major histocompatibility complex genes, then you would theoretically be able to respond to a wider range of diseases. Part of the reason why some people get a really bad case of a disease and other people get a very mild case is going to be their ability to recognize some little bits of protein from whatever that disease-causing organism is. This is true of the flu. It's true of SARS coronavirus 2. It's true of Ebola virus. It's probably true for every virus and every bacterium out there. And yeah, think about it. You know, are you the one in your family who always gets ill, or are you the one in your family that usually, you know, whatever, you don't really notice it? Now, you've probably, if you've been exposed, you've probably been infected. But not everybody who gets infected actually has the same symptoms. Some people are asymptomatic, and that usually means that their MHC and their T cell receptors are doing a very good job. Okay, last little bit of introduction here. Let's talk about these 5 classes of immunoglobulin, 5 antibody classes, or you can call them 5 antibody isotypes. So these are IgM, which is composed of mu heavy chains. Remember, IgM looks like a star, so it's got 10 of those mu heavy chains. And this is the first one that ever appears on B cells. So as B cells are working out what kind of antigen, what kind of antibody they're going to make, the first version they try out is an IgM. And if their IgM is not successful, then the cell dies and another cell takes its place. IgD is another very early one. So this is going to be usually mounted on the surface of mature B cells. And it's a good marker to tell whether you are dealing with a mature B cell as opposed to a B cell that's just trying to figure out exactly what to do. So first, these B cells are going to be IgM positive, and later they're going to be IgM and IgD positive once they're mature. And then they'll keep that IgD. The IgM will go away, and they'll switch to one of these other classes. A typical antibody-producing cell, like a plasma cell, will have switched from IgM, usually to IgG, and then possibly also another step further to IgA or IgE. And as part of this process, it'll make some IgD as well. And these all have the same top of the antibody. So the part that binds the antigen is the same, but the bottom part that controls what kind of cell can bind to the antibody, so the handle part of the antibody, that's the part that is going to be different in every case. And so, yeah, IgM, D, G, A, E. Those are the genes. And IgA is usually secreted in a dimeric form, which means, like I said, that there are it's like 2 IgA molecules joined together back to back. And it's got a little linking chain that joins them up. Okay, so once we get done with this introduction, then we will pause. That'll be the end of this section. But you'll have a good background and a lot of flashcards to make. So get to making those flashcards. Shown up here in the top in panel A, this is an actual, this is the backbone. So just like the simplified version of the protein structure of an antibody. And here what we have are 2 heavy chains. And so the heavy chains are shown in sort of dark bluish purple and in yellow over here. And then we have 2 light chains, and those are shown in pink. The immunoglobulin-like domains are each of these little lumps here. So this pink lump is one immunoglobulin-like domain. This is another one. Here are the immunoglobulin-like domains of the heavy chain 1, 2, 3, 4. So you can see heavy chains have 4 immunoglobulin-like domains. Light chains have 2. So, and that's what they look like. And they do make this sort of, I don't know, it's like an O-shaped structure up here in the top. The part that actually binds to the antigen is going to be somewhere on this surface, usually somewhere around the junction between the heavy chain and light chain area. But you can make antibodies where just the heavy chain binds or just the light chain does the binding, and the other chain is not that important. And then down here, this is the FC portion, the constant region, whereas this is the FAB portion up here, all this that I'm circling, and this is another FAB. And I'll show you how we make those in just a minute. That'll be coming up, I think, in the next section. So this is like a schematic version, and so the colors are going to be the same. So our light chains are going to be in pink here, and there's our little yellow in the back. At this point and this point, the antibody is flexible, so it can wave those little arms around and bind things that are different distances apart and potentially make two bindings onto the same substrate. Since the two heavy chains are going to be identical and the two light chains are going to be identical, that can definitely work. And that is really important for stopping some viruses, for example. You can link to one virus and then link to another virus, and then boom, you basically knocked out two at a time. Shown here at the bottom, this is like a schematic version. These little bars that go in between here, 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 and here, those are disulfide bonds. For each of the different types of heavy chain, there are a different number and arrangement of these disulfide bonds. And I think what you're looking at here is an IgG, because it's got the two disulfide bonds there, and holding the two chains together, and one each holding the light chain on the outside to the heavy chain on the inside. The blue thing here is just a continuation of the chain. Because remember, all proteins are just chains. And so we're showing it very schematically. You can see that it kind of wraps over in 3 dimensions and that they're kind of bendy and awkward looking in reality. All right, so that's a good place for us to pause, and then we will come back and go into a little bit more in antibody terminology and antibody structure.